Welcome back to BioDecode, where biology is decoded into simple, meaningful concepts. This is your trusted audio guide for learning biology the smart and effective way. You're listening to the NCERT Class 11 Biology Audiobook, Chapter 6, Anatomy of Flowering Plants, Exercise Solutions and Key Questions. If you haven't yet listened to the full Chapter 6 audiobook on Anatomy of Flowering Plants, we recommend starting there first. That episode takes you through every concept, from the tissue systems and their organization to the anatomical features of dicot and monocot roots, stems, and leaves. In this episode, we'll go step by step through the NCERT exercise solutions for anatomy of flowering plants, followed by a set of important additional questions to help you revise and strengthen your understanding for both school exams and NEET preparation. Each answer is presented in a detailed and structured manner, precise, factual, and easy to follow, making it ideal for both listening and long-term retention. So, plug in your earphones, focus your mind, and let's begin decoding the internal architecture of plants, from tissues and vascular bundles to the comparative anatomy of roots and leaves. This is BioDecode, and you're listening to Chapter 6, Anatomy of Flowering Plants, Exercise Solutions and Key Questions. Question 1. Draw illustrations to bring out the anatomical difference between a monocot root and dicot root, a monocot stem and dicot stem. Answer. Let us first discuss the differences between monocot and dicot roots. In a dicot root, such as the sunflower root, the outermost layer is called the epiblemma, which bears unicellular root hairs. Beneath it lies the cortex, made up of several layers of thin-walled parenchyma cells with intercellular spaces. The endodermis forms the innermost layer of the cortex, characterized by Casparian strips, deposits of suberin that make the cell walls impermeable to water. Inside the endodermis lies the pericycle, from which lateral roots originate. The vascular bundles are arranged in a radial pattern. Usually, there are 2 to 4 xylem and phloem patches, and between them, a cambium ring develops later. The pith is small, or sometimes absent. In contrast, a monocot root, such as the maize root, also shows epiblemma, cortex, endodermis, pericycle, and vascular bundles. However, there are more than 6 xylem bundles, a condition known as polyarch. Pith is large and well developed, and there is no secondary growth because cambium is absent. The transverse section of a dicot root shows a central small pith with a few xylem and phloem patches arranged alternately along a radius. In contrast, the monocot root shows a larger central pith and multiple xylem and phloem bundles forming a ring-like structure. Now, let us discuss the differences between monocot and dicot stems. A dicot stem has an outer epidermis covered with a thin cuticle. Below the epidermis lies a few layers of collenchymatous hypodermis, providing mechanical strength. The inner cortex is made up of parenchymatous cells with intercellular spaces. The endodermis contains starch grains and is also known as the starch sheath. The pericycle is present above the phloem in the form of semilunar sclerenchymatous patches. The vascular bundles are conjoint, collateral, open, and arranged in a ring. Pith occupies the central region. In a monocot stem, such as maize, the epidermis is single-layered with a thick cuticle and no trichomes. The hypodermis is sclerenchymatous. The vascular bundles are scattered throughout the parenchymatous ground tissue and are conjoint, collateral, and closed. Each vascular bundle is surrounded by a sclerenchymatous bundle sheath, and water-containing cavities are present within them. The pith is not well defined. In the dicot stem, vascular bundles form a circular arrangement with xylem towards the center and phloem towards the periphery, whereas in the monocot stem, vascular bundles appear scattered throughout the section. Now that we have studied the differences between monocot and dicot roots and stems, let us move to the next question, which focuses on the identification of plant stems through their anatomical features. Question 2. Cut a transverse section of a young stem of a plant from your school garden and observe it under the microscope. How would you ascertain whether it is a monocot stem or a dicot stem? Give reasons. Answer. When a transverse section of a young plant stem is observed under the microscope, certain distinct anatomical features help us determine whether it is monocotyledonous or dicotyledonous. If the vascular bundles are arranged in a ring, the stem is dicotyledonous. Such vascular bundles are open because they contain cambium between xylem and phloem, allowing secondary growth. The hypodermis is collenchymatous, and the ground tissue can be differentiated into cortex, endodermis, pericycle, and pith. In contrast, if the vascular bundles are scattered throughout the ground tissue, the stem is monocotyledonous. The vascular bundles are closed, meaning there is no cambium and hence no secondary growth. The hypodermis is sclerenchymatous, and the ground tissue is uniform without differentiation. Additionally, each vascular bundle is surrounded by a sclerenchymatous bundle sheath. In the transverse section of a dicot stem, vascular bundles form a complete ring near the periphery. In the monocot stem, vascular bundles are randomly scattered, giving a characteristic spotted appearance under the microscope. With the knowledge of these structural features, we can now proceed to the next question that involves interpreting vascular arrangement in plant sections. Question 3. The transverse section of a plant material shows the following anatomical features. A. The vascular bundles are conjoint, scattered, and surrounded by sclerenchymatous bundle sheaths. B. Phloem parenchyma is absent. What will you identify it as? Answer. A plant section that shows conjoint, scattered vascular bundles, each surrounded by sclerenchymatous bundle sheaths, and with phloem parenchyma absent, represents a monocot stem. In a monocot stem, vascular bundles are numerous and scattered throughout the ground tissue. Each vascular bundle is conjoint enclosed, meaning xylem and phloem are located on the same radius but separated by no cambium. Each bundle is surrounded by a sclerenchymatous bundle sheath that provides mechanical strength. Phloem parenchyma is absent, and water-containing cavities are present within the vascular bundles. Therefore, based on these anatomical features, the section belongs to a monocot stem, such as maize or sugarcane. After identifying plant organs through anatomical characteristics, we shall now move to understanding specific structures like the stomatal apparatus. Question 4. What is stomatal apparatus? Explain the structure of stomata with a labeled diagram. Answer. The stomatal apparatus is a structural unit in the epidermis of leaves and young stems that regulates gaseous exchange and transpiration. A stoma consists of two guard cells that enclose a stomatal pore. The guard cells are usually bean-shaped in dicots and dumbbell-shaped in grasses monocots. Each guard cell has a nucleus, cytoplasm, and chloroplasts. The inner wall of a guard cell, which faces the stomatal pore, is thicker, while the outer wall is thinner and elastic. Changes in turgidity of the guard cells regulate the opening and closing of the stomatal pore. Sometimes a few surrounding epidermal cells become specialized in shape and size and are called subsidiary cells. The stomatal apparatus thus includes the stomatal pore, guard cells, and subsidiary cells. The diagram of a stomatal apparatus shows two kidney-shaped guard cells enclosing a central pore. In grasses, guard cells appear dumbbell-shaped. Chloroplasts are visible inside the guard cells. The surrounding epidermal cells include subsidiary cells that support the guard cells in their function. Now, let us recall the three fundamental tissue systems found in flowering plants. Question 5. Name the three basic tissue systems in the flowering plants. Give the tissue name under each system. Answer. The three basic tissue systems in flowering plants are: first, The epidermal tissue system. It forms the outermost covering of the entire plant body. It includes epidermal cells, stomata, and epidermal appendages, such as trichomes and root hairs. Second, the ground or fundamental tissue system. This system forms the main bulk of the plant body and includes parenchyma, parenchyma, and sclerenchyma. Depending on the plant organ, it may also include cortex, pith, and pericycle. Third, the vascular conducting tissue system. This system consists of complex tissues, namely xylem and phloem, which are responsible for the conduction of water, minerals, and food. Now, let us move to the next question, which focuses on the significance of studying plant anatomy. Question 6. How is the study of plant anatomy useful to us? Answer. Study of plant anatomy is useful in several ways. First, it helps in understanding the structural organization of plant tissues and organs. Second, it enables us to differentiate between monocot and dicot plants based on internal features. Third, anatomical knowledge aids in identifying adaptations to different environmental conditions, such as xerophytic and hydrophytic structures. Fourth, it helps in taxonomic classification by providing microscopic structural features for identification. Fifth, it also contributes to economic botany, where wood anatomy assists in timber identification and processing industries. Therefore, plant anatomy provides a foundation for both scientific understanding and practical applications in agriculture, ecology, and taxonomy. Finally, let us discuss the internal structure of a dorsiventral leaf. Question 7. Describe the internal structure of a dorsiventral leaf with the help of labeled diagrams. Answer. The dorsiventral leaf, commonly found in dicot plants such as sunflower, shows three main parts: the epidermis, mesophyll, and vascular system. The epidermis covers both upper and lower surfaces. The upper epidermis is known as the adaxial epidermis, and the lower as the abaxial epidermis. Both layers are covered with a cuticle, but the abaxial surface has more stomata. Between the two epidermal layers lies the mesophyll, which is made up of parenchyma cells containing chloroplasts for photosynthesis. The mesophyll is differentiated into palisade parenchyma, consisting of elongated cells arranged vertically and tightly packed beneath the upper epidermis, and spongy parenchyma, composed of loosely arranged cells with large intercellular spaces and air cavities. The vascular system consists of vascular bundles that can be seen in the veins and midrib. The xylem lies towards the upper surface, and the phloem lies towards the lower surface. Each vascular bundle is surrounded by a layer of bundle sheath cells. The transverse section of a dorsiventral leaf shows the upper and lower epidermis enclosing the mesophyll region. Palisade cells appear elongated and tightly arranged below the upper surface, while the spongy parenchyma with air cavities lies beneath them. Vascular bundles are centrally located with xylem facing upwards and phloem downwards. Here are 10 more important and thought-provoking questions from Anatomy of Flowering Plants, questions that often appear in exams and test your conceptual clarity at a deeper level. Take a few moments to reflect on them and try answering in the comments section below. Your participation not only reinforces your learning, but also helps other students who are preparing alongside you. And if you'd like the next audiobook to include detailed explanations for these additional questions, let me know in the comments. Your feedback shapes the upcoming lessons in this series. You've just completed listening to Chapter 6, Anatomy of Flowering Plants, including NCERT, exercise solutions, and extra important questions, here on BioDecode. I hope this audio helped you clearly understand the internal organization of plant tissues, the differences between monocot and dicot structures, and the functional significance of each tissue system. These insights form the foundation for upcoming chapters on plant physiology and reproductive biology, enabling you to connect anatomy with function. So make sure to follow BioDecode and stay tuned for the next part of this NCERT Biology audio series. Keep learning, keep exploring, and I'll meet you in the next audio lecture. NCERT Class 11 Biology, Chapter 7, Structural Organization in Animals. Students, don't forget to like, share, and subscribe to BioDecode for more NCERT audiobooks, quick revisions, and neat preparation guides. See you in the next audio.